Cell, the unit of life. Eukaryotic cells. Eukaryotic cells. The protists, fungi, plants, and animals are included under eukaryotes. Though the presence of membrane-bound organelles, there is an extensive compartmentalization of cytoplasm in eukaryotic cells. An organized nucleus and a nuclear envelope is present in the eukaryotic cells. Eukaryotic cells, in addition, have a variety of complex locomotory and cytoskeletal structures. The genetic material in eukaryotic cells is organized into chromosomes. All the cells in eukaryotes are not identical. Animal and plant cells differ as the plant cell possesses cell walls, plastids, and a large central vacuole which are not present in animal cells. The animal cells, on the other hand, have centrioles which are not present in almost all plant cells. Let us now understand the structure and functions of individual cell organelles. Cell membrane. Only after the invention of the electron microscope in the 1950s, a detailed structure of the cell membrane was studied. The chemical studies conducted on the cell membrane, especially in human red blood cells, helped the scientists to come up with the possible structure of plasma membrane. These studies conducted proved that the cell membrane is made up of lipids which are arranged in a bilayer. Within the membrane, lipids are arranged with hydrophobic tails towards the inner part and the polar head towards the outer sides. This ensures that the saturated hydrocarbons with non-polar tails is protected from the aqueous environment. The membrane's lipid component is mainly made up of phosphoglycerides. Later, it was clearly revealed by the biochemical investigation that the cell membranes are also made up of protein and carbohydrate. In different cell types, the ratio of protein and lipid varies considerably. The membrane of the erythrocyte in human beings has approximately 52% protein and 40% lipids. Membrane proteins can be classified as integral or peripheral depending on the case of extraction. On the surface of membrane lie the peripheral proteins, whereas in the membrane the integral proteins are partially or totally buried. Singer and Nicholson proposed an improved model of the structure of cell membrane which are widely accepted as a fluid mosaic model. According to this model, within the overall bilayer, the quasi-fluid nature of lipid enables lateral movement of proteins. Within the membrane, this ability to move is measured as its fluidity. From the point of view of functions like cell growth, secretion, formation of intercellular junctions, cell division, endocytosis, etc., the fluid nature of the membrane is also important. The transport of the molecules across the plasma membrane is one of the most important functions of the plasma membrane. To some molecules present on either side of it, the membrane is selectively permeable. Across the membrane, many molecules can move briefly without any requirement of energy, and this is called the passive transport. By the process of simple diffusion, neutral solutes may move across the membrane along the concentration gradient, that is, from higher concentration to the lower concentration. From higher to lower concentration, water may also move across this membrane. By diffusion, the movement of water is called osmosis. Through the nonpolar lipid bilayer, the polar molecules cannot pass, and to facilitate their transport across the membrane, they require a carrier protein of the membrane. A few ions or molecules against their concentration gradient are transported across the membrane, that is, from lower to the higher concentration. Such a transport in which ATP is utilized and is an energy dependent process and is called active transport. Example, sodium potassium pump. Cell wall. As you may recall, an outer covering for the plasma membrane of fungi and plants is called the cell wall, which is a non-living rigid structure. Cell wall not only protects the cell from mechanical damage and infection and gives shape to the cell, it also provides barrier to the undesirable macromolecules and helps in cell-to-cell interaction. Other plants have cell wall made of cellulose, hemicellulose, pectins and proteins, while the algae have cell walls made of cellulose, galactans, mannans and minerals like calcium carbonate. The primary wall in the cell wall of the young plant cell is capable of growth which gradually diminishes as the cell matures, and on the inner side of the cell the secondary wall is formed. The middle lamellae holds or glues the different neighboring cells together and is a layer mainly made of calcium pectate. The cell wall and middle lamellae connect the cytoplasm of neighboring cells through plasmodesmata. Endomembrane system. In terms of its structure and function, each of the membranous organelles are distinct, and because their functions are coordinated, many of these are considered together as an endomembrane system. The endoplasmic reticulum, Golgi complex, vacuoles and lysosomes are included in the endomembrane system. The functions of some of the organelles such as mitochondria, chloroplast and peroxisomes are not coordinated with the above components, due to which these are not considered as part of the endomembrane system. The endoplasmic reticulum. A network or reticulum of tiny tubular structures scattered in the cytoplasm called the endoplasmic reticulum are revealed by the electron microscopic studies of eukaryotic cells. The intracellular space is divided into two distinct compartments by the ER, that is, luminal and extraluminal compartments. It is often seen that the ribosomes attach to the outer surface of ER. The endoplasmic reticulum, which have ribosomes on their surface, are called rough endoplasmic reticulum. When the ribosomes are absent on the surface of the endoplasmic reticulum, they appear smooth and are called smooth endoplasmic reticulum. In the cells actively involved in protein synthesis, the secretion RER is frequently observed. With the outer membrane of the nucleus, the RER are continuous and extensive. The major site for the synthesis of lipid is the smooth endoplasmic reticulum. Lipid-like steroidal hormones are synthesized by SER in animal cells. Golgi apparatus. Densely stained reticular structures was first observed by Camillo Golgi near the nucleus. After him, these were later named as Golgi bodies. Many flat disc-shaped sacs or cisternae of 0.5 μm to 1.0 μm diameter constitute the Golgi bodies. The Golgi bodies are staked parallel to each other. In Golgi complex, varied number of cisternae are present. Near the nucleus, the Golgi cisternae are concentrically arranged with distinct convex cis or the forming phase and concave trans or the maturing phase. Entirely different but interconnected are the cis and the trans phases of the organelle. The function of packaging materials are principally performed by the Golgi apparatus to be delivered either to the intracellular targets or secreted outside the cell. In the form of vesicles, the materials to be packaged from the ER fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. Due to this, the Golgi apparatus remains in close association with the endoplasmic reticulum. On the endoplasmic reticulum, a number of proteins synthesized by ribosomes are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. The important site of formation of glycoproteins and glycolipids is Golgi apparatus. Lysosomes. Membrane-bound vesicular structures called lysosomes are formed by the process of packaging in the Golgi apparatus. Optimally active at the acidic pH are the isolated lysosomal vesicles, which have been found to be very rich in almost all types of hydrolytic enzymes. Carbohydrates, proteins, lipids, and nucleic acids can be digested by these enzymes. Vacuoles. The membrane-bound space found in the cytoplasm is the vacuole. Water, sap, excretory product and other materials not useful for the cell are present in the vacuole. A single membrane called tonoplast is bound over the vacuole. Up to 90% of the volume of the cell is occupied by the vacuole in plant cells. The tonoplast facilitates in plants the transport of the number of ions and other materials against concentration gradients into the vacuole, due to which their concentration is significantly lower in the cytoplasm than in the vacuole. For excretion in amoeba, the contractile vacuole is important. By engulfing the food particles, food vacuoles are formed in many cells as in protists. Mitochondria. Under the microscope, unless specifically stained, mitochondria are not easily visible. Depending on the physiological activity of the cells, the number of mitochondria per cell is variable. A considerable degree of variability is also observed in terms of shape and size. It is typically sausage-shaped or cylindrical with a length of 1.0 to 4.1 μm and a diameter of 0.2 to 1.0 μm. Each mitochondrion with the outer membrane and the inner membrane is a double membrane bound structure dividing its lumen distinctly into two aqueous compartments, that is, the outer compartment and the inner compartment. The matrix forms the inner compartment. The continuous limiting boundary of the organelle forms the outer membrane. A number of infoldings called the cristae form the inner membrane towards the matrix. The surface area is increased by the cristae. Associated with the mitochondrial function, the two membranes have their own specific enzymes. The sites of aerobic respiration are the mitochondria. They are called powerhouses of the cell as they produce cellular energy in the form of ATP. A single circular DNA molecule, a few RNA molecules and ribosomes are present in the matrix along with the components required for the synthesis of proteins. The division of mitochondria takes place by fission. Plastids. In all plant cells and in euglenoids, the plastids are present. As the plastids are large, they are easily observed under the microscope. They impart specific colors to the plant as they bear some specific pigments. Plastids can be classified based on the type of pigments into chloroplasts, chromoplasts, and leucoplasts. Chlorophyll and carotenoid pigments are present in the chloroplast and they are responsible for trapping light energy essential for photosynthesis. Carotene, xanthophylls and other fat-soluble carotenoid pigments are present in the chromoplasts. Thus, a yellow, orange or red color is given to the part of the plant. The colorless plastids of varied shapes and sizes with stored nutrients are the leucoplasts. Amyloplasts store carbohydrates, example potato. Elaioplasts store oils and fats, whereas the eleuroplasts store proteins. In the mesophyll cells of the leaves are present a majority of the chloroplasts of the green plants. Chloroplasts have variable length, width, and they are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles. Their number varies from 20 to 40 per cell in the mesophyll to 1 per cell of the Chlamydomonas, a green alga. The chloroplasts are also double membrane bound like mitochondria. The inner chloroplast membrane is relatively less permeable out of the two membranes. The stroma is the space limited by the inner membrane of the chloroplasts. In the stroma, a number of organized flattened membranous sacs called the thylakoids are present. In stakes like the piles of coins called grana or the intergranal thylakoids are arranged the thylakoids. In addition, connecting the thylakoids of the different grana are the flat membranous tubules called the stroma lamellae. A space called the lumen is enclosed by the membrane of the thylakoids. Enzymes required for the synthesis of carbohydrates and proteins are present in the stroma of the chloroplast. Small double-stranded circular DNA molecules and ribosomes are present in the stroma of the chloroplast. In the thylakoids, chlorophyll pigments are present. The chloroplast ribosomes are smaller than the cytoplasmic ribosomes. Ribosomes. Under the electron microscope, the granular structures, the ribosomes, were first observed as dense particles by George Pallad. The ribosomes are not surrounded by any membrane but are composed of ribonucleic acid and proteins. The prokaryotic ribosomes are 70S while the eukaryotic ribosomes are 80S. The S stands for the sedimentation coefficient. It is indirectly a measure of size and density. The ribosomes, both 70S and 80S, are composed of two subunits. Cytoskeleton. In the cytoplasm, an elaborate network of filamentous proteinaceous structures, collectively referred to as cytoskeleton, is present. In a cell, the cytoskeleton are involved in many functions such as motility, mechanical support, maintenance of the shape of the cell. Cilia and flagella. Hair-like outgrowths of the cell membrane are the cilia and flagella. Causing the movement of either the cell or the surrounding fluid are small structures, the cilia, which work like oars. Comparatively longer are the flagella, which are responsible for cell movement. The flagella are also present in the prokaryotic bacteria, but from the eukaryotic flagella they are structurally different. The cilium or the flagellum show that they are covered with plasma membrane under the electron microscopic study. A number of microtubules running parallel to the long axis are present in their core called the axoneme. The axoneme usually has a pair of centrally located microtubules and 9 pairs of doublets of radially arranged peripheral microtubules. An arrangement of such axonemal microtubules is referred to as the 9+2 array, connected by bridges and also enclosed by a central sheath are the central tubules, which is connected to one of the tubules of each peripheral doublets by a radial spoke. Thus, 9 radial spokes are present. Also interconnected by linkers are the peripheral doublets. From centriole-like structure called the basal bodies emerge both the cilium and flagellum. Centrosome and centrioles. An organelle centrosome usually contains two cylindrical structures called centrioles. Amorphous pericentriolar materials surround the centrosome. Perpendicular to each other lie both the centrioles in a centrosome in which each has an organization like the cartwheel. They constitute 9 peripheral fibrils of tubulin which are evenly spaced. Each peripheral fibril is a triplet. Also linked are the adjacent triplets. The proteinaceous central part of the centriole is called the hub, which connects the tubules of the peripheral triplets by radial spokes which are made of protein. The basal body of cilia or flagella and spindle fibers form the centrioles and give rise to spindle apparatus during cell division in animal cells. Nucleus. As earlier as 1831, nucleus was first described by Robert Brown as a cell organelle. Later, Fleming gave the name chromatin to the material of the nucleus stained by the basic dyes. The nucleus of the interphase has elaborate and highly extended nucleoprotein fibers called chromatin, nuclear matrix, and one or more spherical bodies called nucleoli. It was revealed from the electron microscopy that the nuclear envelope, which consists of two parallel membranes with a space between called a perinuclear space, forms a barrier between the cytoplasm and that of the materials present inside the nucleus. Usually, the outer membrane of the nucleus remains continuous with the endoplasmic reticulum, which also bears ribosomes on it. Minute pores interrupt the nuclear envelope at a number of places which are formed by the fusion of its two membranes. These pores of the nucleus form the passages through which movement of protein molecules and RNA takes place in both directions between the cytoplasm and the nucleus. Per cell normally there are only one nucleus, and also frequently observed are the variations in the number of nuclei. Some mature cells do not have nucleus, example the erythrocytes of many mammals and sieve tube cells of vascular plants. The nucleolus and chromatin are present in the nuclear matrix or the nucleoplasm. The spherical structures present in the nucleoplasm are the nucleoli. With the rest of the nucleoplasm, the content of nucleolus is continuous as the nucleolus is not a membrane-bound structure. At the nucleolus, active ribosomal RNA synthesis takes place. In the cells actively carrying out protein synthesis, larger and more numerous nucleoli are present. You may recall that a loose and indistinct network of nucleoprotein fibers called chromatin are present in the interphase nucleus. But during different stages of cell division, in place of the nucleus, cells show structured chromosomes. DNA and some basic proteins called histones, some non-histone proteins, and also RNA are present in the chromatin. Distributed among its 46 chromosomes, approximately 2-meter long thread of DNA is present in a single human cell. A primary constriction or the centromere, on the sides of which disc-shaped structures called kinetochores are present essentially in every chromosome. The chromosome can be classified into 4 types based on the position of the centromere. A middle centromere with two equal arms of the chromosomes is present in the metacentric chromosome. A chromosome with a centromere nearer to one end of the chromosome, resulting into one shorter arm and one longer arm, is seen in a submetacentric chromosome. An acrocentric chromosome has a centromere which is situated close to its end, forming one extremely short and one very long arm, while a terminal centromere is present in a telocentric chromosome. Sometimes at a constant location, a few chromosomes have a non-staining secondary constriction which gives the chromosome the appearance of a small fragment called the satellite. Microbodies. In both plants and animal cells, many membrane-bound minute vesicles that contain various enzymes called microbodies are present. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content.